Hello and welcome to my next video where I'm doing the OCR F214, that's the Unit 4 biology paper from January 2013. A few people asked me to do some of these, so I'm doing Unit 4.1 now. Please feel free to do the paper beforehand. I'll give a chance to pause so you can go off and do it, and then come back and we'll go through the mark scheme. In this paper I have done it myself using the mark scheme to make sure I don't make any mistakes. And I've also added in extra points and counted them as marks. So I haven't given, so for example, if it's a 4-mark question but there are 7 points in the mark scheme, I might have put all 7 points in and tick them so you can see where the marks are allocated. So feel free to pause now and come back once you finish the paper. Right, good. And those who don't want to do the paper, that's fine, we can have a look at it anyway. So, question 1, nice and simple. Figure 1.1 represents a sensory neuron connected to its associated receptor cells. Identify the parts of the neuron labelled A to D. Now an A is at a receptor cell going to a dendron, so it's probably dendrite. It is. The long bit to a cell body from receptors, you should know, is a dendron. The bit with the nucleus is clearly the cell body, and the bit going away from a cell body is always an axon. Now what is represented by the arrows? Well, that's the direction of transmission of a nervous impulse. Now first, these are the first 5 marks, very easy, just simple recall facts. But then they give you a straight out, here you go, here is a quality of written communication question. Now remember, in Unit 4, just like Unit 1, quality of written communication is given for 3 technical terms spelt correctly in each question it appears in, which will be 2. So it says, describe and explain how the resting potential is established and how it is maintained in a sensory neuron. Now the first thing I think of whenever I see resting potential is always the 3 and 2. You have 3 sodium out for 2 potassium in. So remember that. The sodium potassium pump uses ATP because it's active, it's a pump, to actively transport 3 sodium ions out of the neuron and 2 potassium ions into the neuron. That one statement there has given me 3 marks. I would have got an extra one if, well, I can't get an extra one until I've used 3 technical terms which haven't been used here, but I've got my 3 marks. Because it says the sodium potassium pump uses ATP to actively transport, 1 mark. You have to mention the sodium ions, 3 sodium and 2 potassium to get 1 extra mark, but also the fact you've mentioned the numbers means you get a second. So you get 3 marks for that. Potassium channels are open and potassium ions diffuse out of the neuron. So it's important to say that they diffuse out of the neuron. Voltage gated sodium ion channels are closed. So it's important here to mention whatever happens to one thing, mention what happens to the other. So are potassium ions channels open or closed? Well, they're open. So what can happen? Well, they can diffuse out of the neuron. But the sodium ones are closed. So the membrane is less permeable to sodium ions, so less can diffuse back in, meaning you've got that constant gradient. And in here, there is a number of options for quality of written communication, but I chose sodium potassium pump and diffuse. Continuing in the question, another simple recall fact. Figure 1.2 shows the changes in the membrane potential of a sensory neuron when the receptor cells are stimulated. Figure 1.3 shows the strength of the stimuli that result in the corresponding changes in membrane potential. Now, I'm going to go through this before looking at the questions. Now, first we can see small stimuli and a little nervous impulse but not an action potential. Stronger stimuli and an action potential has been generated. An even stronger stimulus and exactly the same action potential. An even stronger stimulus than that, exactly the same action potential. So you can probably tell it's going to be something about the all or nothing response. We've also got X, Y and Z. State the term used for each of these. X is clearly depolarization, Y is repolarization, Z is hyperpolarization. Yet again, very simple recall questions. What term is used to refer to the value of minus 50 millivolts? Well, that's the threshold potential. And that's just things you have to learn. Comment on the relationship between the strength of the stimulus, as shown in figure 1.3, and the resulting action potential, as shown in figure 1.2. As I said, all or nothing. So you want to make sure you've got that in there. So, only stimuli that reach the threshold potential become an action potential. All action potentials are the same size regardless of the strength of the stimulus, and this is called the all or nothing response. So if you just remember all or nothing response, you've got 1 mark there, and then just explaining it gives you another mark. Question 2, I think this is one of the harder questions on the paper. The liver is an organ that is metabolically very active, carrying out over 500 different functions. Some of its important functions include converting chemicals, including toxins, into other compounds. Figure 2.1 outlines some of the reaction pathways that take place in the liver cells. The underlined words represent toxic compounds. So here we know ammonia is toxic, ethanol is toxic, and lactate is toxic. So yet again, another 2 very simple recall questions. State the product of the ornithine cycle in pathway P and the organ which this product is transported to for removal from the body. Now you just remember it's urea, and since urea it must be kidneys. The lactate enters pathway S produced by cells such as muscle cells undergoing anaerobic respiration. Suggest why this lactate is converted into pyruvate by the hepatocyte liver cells rather than by respiring cells in which it is produced. Now we've been told lactate is toxic. Now the reason why they've told us that is because it's going to be used. Lactate is toxic. And what's important about it being toxic? Well, surely if it's toxic, it's going to be, damaged by both of them? Well, no, liver cells have a much greater resistance. They can tolerate lactate. Also, if you think about what else could be to do with lactate, well, lactate is a chemical, it must be broken down. What do you need to break down a chemical? An enzyme, perhaps only the liver has the correct enzymes, and indeed it does. So just remember that. Lactate, we've been told it's toxic, so that's going to definitely be used in the question somewhere. They wouldn't have told us it. And also, since it is a chemical and it's broken down, enzymes must be involved. There will be a question on this paper, or Unit 5, which involves enzymes somewhere, because enzymes are so important. Now, insulin only remains in the bloodstream for a relatively short time. Pathway R breaks down insulin in the liver. Explain what might happen if the liver did not break down insulin. Well, what does insulin do? It lowers blood glucose levels. Well, that would mean blood glucose concentration would fall too low. And then just explain it. Glucose will continually be converted to glycogen, and glucose will be taken up by the cells. Less glucose will be available then in the blood, so we wouldn't have much respiration, so not enough ATP will be made, and this would eventually lead to death. Yes, you can just answer death/coma and you'll get a mark. Now, alcohol is oxidized in the liver by pathway Q. If a person has a high alcohol intake, it will result in the production of excess reduced NAD. Excess reduced NAD in the liver cells will influence some metabolic pathways by inhibiting the conversion of lactate to pyruvate, inhibiting fatty acid oxidation, and promoting fatty acid synthesis. Now, looking at this, these 3 things we've been told, we've been given information again. Inhibits the conversion of lactate to pyruvate. As we said, lactate is toxic, pyruvate isn't. So, something about toxicity again. Inhibiting fatty acid oxidation and promoting fatty acid synthesis means you have more fatty acids. Fatty acids are deposited in the liver, and this causes cirrhosis. So instantly there, you've got 2 ideas, and you can almost certainly tell that's what they're going to want. Using this information, and the information in figure 2.1, suggest the consequences for the liver metabolism if a person had a regular high alcohol intake. Well, we know that a buildup of lactate will kill liver cells, because even though they are more tolerant to it, it will eventually kill them. That's 1 mark. Now, deamination won't be able to occur because you need your NAD, you need to be able to oxidize. And fatty acids will be deposited in the liver, resulting in cirrhosis, as I said earlier. And finally, state precisely where in the liver cell the excess reduced NAD can be reoxidized. Well, basically we've got reduced NAD. Reduced NAD has 2H. 2H can then be dropped off, and so you lose 2H. Where in the... Well, what process do we know about where you lose 2H? Well, there's only one, and that's oxidative phosphorylation, which occurs in the cristae of mitochondria, or the inner membrane. So relatively simple. It's simple if you think about it, but it requires a lot of thought. Now, explain what's meant by the terms autotroph and heterotroph, 2 definitions. Autotroph can synthesize large organic molecules from inorganic molecules. Heterotrophs can ingest and digest complex organic molecules from other organisms. Figure 3.1 is a transmission electron micrograph showing that part of a chloroplast, including some of the internal membranes. Now we've looked at E. Now, if you ever have that thing where there's a big stack of something with that kind of open bracket, it's going to be a grana. And F is pointing to nothing in particular, which means it's a stroma. The chloroplast contains fat droplets as shown in figure 3.1. These act as a reserve of raw material for the chloroplast. Suggest what this raw material might be used for in a chloroplast. Well, what can fats be used for? Fatty acids, and for phospholipids, which are fats. So that's membrane formation. Again, a little bit of thought. Here's the other quality of written and communication question. Figure 3.2 represents the light harvesting system found in the surface of the internal membranes of the chloroplast. Using the information in figure 3.2 to describe how light is harvested in the chloroplast membranes. Basically, it wants the stage from light going into the photosystem to when it actually reaches the reaction center. So, firstly, state what all those little sections are. The primary and accessory pigments are the circles are found in the photosystem, so they know what you know what we're talking about. Light energy, also known as photon energy, is absorbed by the accessory pigments, exciting the electrons, thus moving it to a higher energy level, or energy state. This energy is passed from one pigment to another until it is passed onto the reaction center, which contains chlorophyll A. That's all you need to say. You just basically need to say the light is passed along from one accessory pigment to another, and what happens to the electrons. And that would be simple if you've learnt it, but it's very easy to overlook this section, so it could be quite a hard question for some. Now, here's the kind of you'll always get a bit of an experiment at some point. Here's the experiment bit. Many herbicides act by inhibiting photosynthesis in weeds. A series of research studies were carried out to evaluate the effectiveness of a triazine herbicide on the yield of a crop of corn, Zea maize. Some of the data obtained is shown in figure 3.1. Calculate the yield difference caused by the application of herbicide in study G. So basically it wants the two figures that have been left out from the table. Now what I do here is I look at the previous example. I've used, which example have I used? Oh, I haven't actually written it down here. Let's say A. What you do is you look at and you go, well, let's have a look. We've got kilograms per, I think that's hectare minus, per hectare, is plus 435.5. So it's positive, which means the bigger number must have the smaller number taken away from it. So in this case, it is herbicide minus not herbicide. So I do 10807.1 minus 11671.4. So I've taken herbicide away from non-herbicide, and that gives that value. Now, in terms of the percentage, I look again. Let's have a look at A again. Well, this is just trial and error. I tried putting 435.5 divided by 8321.4 times by 100. That didn't come out with a good value. That didn't come out with the correct 5.2%. So I try 435.5 divided by 8756.9 times by 100, and that does come out the correct value. So there's only three numbers you've got in each table, so it must be one of them. So therefore, I do my previous value of minus 864.3 over 10807.1 times by 100. I get a value of minus 7.4%. So it's just trial and error and practice. You'll see how to do these questions. Suggest why the researchers concluded that the data obtained from study E was not useful in evaluating the effectiveness of the herbicide. Well, if we have a look at E, firstly we see it's a very big difference, so it's not a very accurate one compared to all the others. We then have a look at the number of plots. 9, 21, 30, 20, 2, 66 and 17. There's not enough examples, so the number of plots tested was very small. Now, triazine herbicide acts on the weeds by binding to a specific protein associated with photosystem 2, blocking the movement of electrons between electron carriers. Explain the effect that the herbicide binding to this protein will have on photosynthesis. Photosystem 2 only used in non-cyclic photophosphorylation. So we know it's going to prevent non-cyclic photophosphorylation, but cyclic photophosphorylation will occur and produce ATP. That's 2 marks. Now, also what will happen, non-cyclic photophosphorylation, no NADP can be reduced. There's no electrons are available, so overall you can have no Calvin cycle. So photosynthesis will effectively stop. Now, plants treated with triazine herbicide can, when illuminated under experimental conditions, be seen to be fluorescent, emit light, and given off small quantities of heat. Just how this experimental finding can be explained. Heat and light, both forms of energy, so energy must be given off. Now the next question, I like this question, and I've actually explained it. I don't normally explain it in the actual paper, but I have. So basically, here are some biological terms. Select from the turn above the terms that refer to each of these. So we have glucagon, gluconeogenesis, glycogen, glycogenesis. We have glyconeolysis, glycolysis, insulin, negative feedback. So they're all very similar words. So select from the list above the term or terms that refer to a stage in respiration. It says a stage is going to be one, and that's the easy one, glycolysis. Hormones. Now again, you should know it's glucagon. Don't get confused with glycogen. It's not glycogen, it's glucagon and insulin. Another relatively easy one. Now the next one. Now this is where, if you can look at how the word works, it's quite useful, and you've seen I've used it at the bottom. So, processes that produce glucose. And the three big ones to break down is gluconeogenesis, glycogenesis, and glycogenolysis. I'm going to go through each of them. I'll go through one example here. Gluconeogenesis. Gluco means glucose. Neo means new, and genesis means creation or formation of. So we have glucose new formation, or in other words, the formation of new glucose. So, processes that produce glucose. Well, does this produce new glucose? Yes, it does. That's an answer. Next one. Glycogenesis I'm going to look at next. Glycogenesis, that means glycose, glycogen, genesis is formation of. So it's the formation of glycogen from glucose, because that's how glycogen is made. So, is glycogen glucose? No, it isn't. It's a chemical made up of glucose, but it doesn't produce glucose, so that's not a correct answer. And finally, glycogenolysis. Glycogen, again, is, well, glycogen. Lysis is breaking up of. So the breaking up of glycogen, what do you get when you break up glycogen? You get glucose. So it produces glucose. So the answer is gluconeogenesis, the formation of new glucose, and glycogenolysis, the breaking up of glycogen to form glucose. And then finally, processes that have glucose as a starting point, or glycolysis does, if you remember. Started with glucose, and also glycogenesis, because we said that's the formation of glycogen from glucose. So if you kind of learn if you can break up those words and see what each of them mean, this question becomes quite easy and actually quite fun. Next question, quite an easy bit. Adenosine triphosphate, ATP, is an important product of respiration. The ATP molecule is made up of 5 subunits. 3-phosphate adenine, and ribose. Now in the space below, show these joined together. Basically, ribose connects one phosphate and adenine, and then the phosphates connect to the other 3. Just look at the diagram and learn it. So here's the type of reaction that removes a phosphate group from an ATP molecule. Well, a bit is being removed, so it's being hydrolyzed, the breaking of a chemical bond. The formation of ATP is now widely accepted as being achieved by the process of chemiosmosis. Various pieces of evidence have been documented to support this theory. 3 of these are described below. I'm not going to read them out, but you can read them for yourselves, but I will go through it. So, is it 1, 2, or 3, or none of that actually have this as evidence? Electron transfer occurs on the inner membrane of the mitochondrion. So, electron transfer. Now, we're looking for something that's to do with the membrane. The isolated mitochondrial membrane, number 1, this is, that have had their outer membranes moved, electron transfer takes place, but the mitochondria are unable to produce ATP. Now what it's saying is electron transfer occurs, but even with so it must happen on the inner membrane because it doesn't need the outer membrane to occur. Next one, protons are actively pumped across the inner membrane, mitochondrial membrane, into the intermembrane space. Now, only 2 actually really talks about pH, importantly. Actually, no, so there's 3. So 2 and 3 both talk about it. So we're looking at those 2. The pH of the intermembrane space is lower than the pH inside the rest of the mitochondria. Okay? Well, that's just saying that there's more protons in the intermembrane space than in the matrix. Now that means nothing. That doesn't actually prove that. You could say, well, they link, but no, they don't prove one another. And the outer mitochondrial membrane is permeable to protons. If isolated, mitochondria spider, that's talking about the outer membrane, that doesn't prove it at all. So nothing proves that statement. And finally, protons accumulate in the intermembrane space. Now you can say 2 or 3 here, because the pH in intermembrane space is lower. So obviously, more protons. That's definitely proof. Now, the outer membrane is permeable to protons. If isolated, mitochondria are supplied with ATP and inorganic phosphate and placed in a solution of pH 8. No ATP is produced. If, however, these mitochondria are placed in an acidic solution, ATP is produced, clearly showing that protons move into the intermembrane space, so that is also proof. And last question now. There's always a fill in the blanks. Complete the passage using the most suitable term in each case. The blood in the glomerulus has a high pressure. Well, it's always hydrostatic. It just is. Which forces small molecules such as glucose and, well, what else? Amino acids. There's a lot of examples. Amino acids, water, ions. Look at the mark scheme for all the others. I just chose amino acids. Out of the glomerulus and into the lumen of the Bowman's capsule. This process is known as, well, there are only two processes that occur in the kidneys, and that's ultrafiltration and selective reabsorption. And selective reabsorption happens in the loop of Henle/collecting duct, so this is ultrafiltration. In the proximal convoluted tubule, the glucose, most of the and some of the salts are reabsorbed into the blood. Well, it's water. Most of the water is reabsorbed. And into the blood, what surrounds the nephron? Well, it's always the capillaries. That's just learning the process. One aspect of the kidney's homeostatic role is the ability of antidiuretic hormone ADH to increase the number of aquaporins in the plasma membrane of the cells lining the collecting duct. This increases the amount of water reabsorbed. ADH is released in response to a decrease in the water potential of the blood plasma. State precisely where the cells that detect a decrease in water potential of the blood are found. Well, anything to do with homeostasis, anything, is always the hypothalamus. Always the hypothalamus. I think in this case, ADH is actually released from the posterior pituitary gland, but it is detected in hypothalamus. Hypothalamus is the homeostatic centre. Name the centres that detect this decrease. Well, it's osmoreceptors because they deal with water potential. Here we have a flowchart. Name the part of the adrenal gland that releases aldosterone. Now, you probably haven't heard of aldosterone. In Figure 6.1 outlines some of the events that take place if the blood volume decreases. So normal blood pressure, significant blood loss, decrease in blood pressure. Receptors detect this decrease. Adrenal glands stimulated to produce aldosterone, a mineral allocorticoid. And aldosterone promotes the uptake of sodium ions from the filtrate in the distal convoluted tubule. You just have to know that the medulla always produces adrenaline. And aldosterone is a steroid. And steroids are produced in the cortex. That's a hard question though. Suggest and explain what effect the action of aldosterone will have on the secretion of ADH. Well, aldosterone promotes the uptake of Na+ from the filtrate in the distal convoluted tubule, so into surrounding, lowering the water potential of the blood, which means more ADH is released to increase the water potential of the blood. And finally, a nice easy question is state the name of the mechanism that results in the decrease in aldosterone secretion, negative feedback. It's about time that mark came up. So, I hope this helped. Any questions, as always, please ask. Comment, like if you actually enjoyed this and this was helpful. So thank you for watching, and goodbye.